The overall goal of the following experiment is to determine relative changes in tRNA charging levels. This is achieved by isolating RNA at a low pH before and after treatment to isolate tRNA with intact charging levels. As a second step, the RNA is subjected to a periodate oxidation treatment, which allows for determination of relative charging levels. Next, ligate samples to a fluorescent oligonucleotide and hybridize on a microarray in order to measure changes in ligation levels due to tRNA charging. Results are obtained that show genomic level changes in tRNA charging based on tRNA microarray technology.
Hi, I'm John Zaborske from the laboratory of Dr. Tao Pan in the Department of Biochemistry and Molecular Biology at the University of Chicago. Today I'll be showing you a procedure for isolating and measuring charged tRNA at the genomic level. We use this to measure changes in tRNA charging during stress response in our lab. So, let's get started.
To isolate total charged tRNA, start by growing a minimum of the equivalent of 30 OD600 of yeast cells, which should produce 30 micrograms of total RNA. Pellet the cells in a tabletop centrifuge. Resuspend cells in a lysis buffer solution. Add an equal volume of sodium acetate saturated phenyl chloroform and vortex for 30 seconds, followed by icing for at least 30 seconds. Repeat vortexing and icing 2 more times. Centrifuge the sample at 18,600 RFC for 15 minutes at 4 degrees Celsius. Then remove the aqueous layer and perform another acetate saturated phenyl chloroform extraction. After the second extraction,